While the ability of small interfering RNA to silence disease-causing genes specifically renders them as a class of powerful drug candidates, delivering them to sites of therapeutic action is a challenge. Practically feasible synthetic carriers for systemic delivery of siRNA in humans have to be precisely assembled with predetermined sizes, surface properties, site-specific responsiveness, and reproducibility. In their article in Advanced Functional Materials, Professor Tuo Jin and colleagues from Shanghai Jiao Tong University address the issues above by constructing a synthetic carrier of siRNA with customizable size, pH-responsive degradability, and optimized surface population of cell-targeting agent, using a series of thermodynamically self-regulated assembling processes. A shift-based reaction between linear spermine, imidazole, imine oligomers and branched low molecular weight polyethylenimine forms networked polyamines in water. The size of this networked cationic polymer is controlled by the zeta potential of the growing polymer. Increasing the pH of the solution from 9 to 12, or more conveniently adding sodium chloride from 0 to 30 millimolar in solutions of pH 9, results in a gradual increase in the polymer's diameter from 140 nanometers to over 400 nanometers. Each single molecule of the size customized cationic polymer can pack sufficient copies of siRNA inside its network cavity before collapsing electrostatically to a dense unimolecular polyplex of reduced diameter. The polyplex can be functionalized by adding a tri-block copolymer, where the distal end of its PEG block can be modified with a pentafluorophenol ester for hooking cell-targeting agents. In this way, the copolymer can be conjugated with a tumor-targeting agent, RGD. The resulting nanoparticle is called a polyraplex, which, having the right surface population of cell-targeting agent, can be absorbed onto the target cells. As an example, mice implanted with U87 tumour cells were injected with a cyanine 5 siRNA-loaded RGD polyraplex. Fluorescence intensity increased dramatically in the tumour when compared to naked cyanine 5 siRNA and the RGD-free polyraplex. Notably, in mice administered with anti-VEGF siRNA, tumours could be reduced to 31.5% of their original size using the RGD polyraplex. To find out more about this thermodynamically assembled multi-purpose drug delivery system for siRNA, please visit the Advanced Functional Materials homepage.